Good day, this is the Professor, CBMG PhD, and today's Friday, March 13th, 2026. It is 5:58 and 16 seconds, p.m. Eastern Standard Time, and today's research, starving cancer cells. So let's hop to it here. A mere molecule can starve cancer cells without harming healthy cells. A mere image amino acid may offer a surprisingly simple way to stall aggressive cancers without harming healthy cells. Date March 12, 2026. Source: University of Geneva. The scientists have discovered that a rare mirror image version of the amino acid cysteine can dramatically slow the growth of certain cancers while leaving healthy cells largely untouched. Unlike most anti-cancer treatments that harm normal tissues, this molecule, called D-cysteine, is taken up mainly by some cancer cells through a specific transporter on their surface. Once inside, it shuts down a crucial mitochondrial enzyme that cancer cells rely to produce energy to maintain DNA, effectively halting their growth. Amino acids are small molecules that serve as the building blocks of proteins. They connect together like beads on a string to form the proteins needed by living organisms. There are 20 amino acids used to construct the proteins found in all forms of life. These two molecules exist in two versions known as L-laboratory and D-dextrorotatory. The two forms are mirror images of one another, similar to the difference between a person's left and right hands. Although they have the same chemical components, their three-dimensional structures differ. Human biology relies almost entirely on the L forms to build proteins, while the D forms are rarely used. The scientific research has explored how various amino acids influence cancer cell growth. Their experiments revealed that the D version of cysteine D-cis, which contains a sulfur atom, can strongly suppress the growth of certain cancer cells. In laboratory experiments, healthy cells, however, were not affected. And this difference between cancer cells and healthy cells is easily explained. D-cis is important in the cells via a specific transporter that is present only on the surface of certain cancer cells. In fact, we observe that if we express this transporter on the surface of healthy cells, those cells stop proliferating in the presence of D-cis. It blocks an essential enzyme called NFS1, located in the mitochondria, the cell's powerhouses. This enzyme plays a key role in producing iron-sulfur clusters, small structures that are indispensable for many processes, such as cellular respiration, DNA and RNA production, and maintaining genetic integrity. When NFS1 is blocked, several critical cellular functions break down. Cancer cells experience reduced respiration, DNA damage increases, and the cell cycle stops. Together, these effects prevent the cells from continuing to grow and divide. To test whether this approach could work in living organisms, the scientists treated mice that had aggressive mammary tumors that are typically difficult to treat. The results were promising. Tumor growth slowed significantly, and the animals did not show major side effects. This is a very positive signal. We now know it is possible to exploit this specificity to target certain cancer cells. However, we still need to determine whether D-cis could be administered at effective doses in humans without causing harm. If further studies confirm its safety and effectiveness in people, D-cysteine could become a relatively simple and selective therapy for cancers that produce high levels of the transporter responsible for bringing the molecules into cells. The strategy may also help prevent metastasis, a crucial stage in cancer progression. And this video was created for educational purposes only. My code name is Will. I'm Professor Christopher Brunet, leader of research, scientist ufologist, psychic computer technician, computer programmer, philanthropist writer, mathematician artist, photographer film producer, chef animal nature lover. My passion is world peace. Have a great day. Good day. This